Hi everyone. My name is Tatiana Kuzmenko, and I am one of the instructors for the General Biology Lab course at Loyola Marymount University in Los Angeles. The goal of this course in the fall semester is to introduce students to a variety of life forms while emphasizing biodiversity. At the same time, we also wanted to introduce students to the research process through inquiry-based lab activity, where working in groups, they form hypotheses, design experiments, collect and analyze data, and share results with their peers. The fungi bioremediation experiment was developed 2 years ago in response to a gap in students' knowledge about fungal kingdom and appreciation its role in human and environment. And our second goal was also to connect our students better to real-world environmental challenges like pollution and waste management. Mushrooms, like you may know, take time to grow. So we run this experiment as a side project, launching it at the beginning of semester and collecting the data at the end, after about 14 weeks. We chose oyster mushrooms as a model organism because they are safe, easy to grow, and remarkably effective decomposers. Students track the development of the mycelium through the glass containers each week, and at the end of the project, we open the jars in the humidity chambers and let the mycelium fruit and produce actual mushrooms before digging in and examining the decomposed materials. And this final stage always generates a lot of excitement among students. Jack, who is going to present the work, did it as a student the first year uh, of the experiment, and then later he taught it as a second year as a TA. He was so inspired by this project that he conducted a meta-analysis of all student group outcomes and identifying broader trends and patterns in fungal decomposition. So in NABT, at an NABT conference this fall, I will share the full setup and instructions how to implement this in your classrooms. But today, Jack will walk you through the experiment from the student's perspective and share the fascinating decomposition results he and his peers observed. So welcome, Jack.
Thank you for joining us today. My name is Jack Spiecker, a junior at Loyola Marymount University, and today I'm excited to share our classroom study on bioremediation using oyster mushrooms. Our title, Waste to Wonder, Substrate-Dependent Bioremediation Revealed in a Classroom Study Using Oyster Mushroom, Pleurotus austriatus, reflects our key finding. Oyster mushrooms can decompose a wide range of waste materials, but success depends entirely on substrate type. Let's dive in. Our main finding is that oyster mushrooms can decompose a wide range of waste materials, with degradation outcomes strongly shaped by the type of substrate. As shown in Figure 2, the 5 categories of substrates varied significantly in their levels of decomposition. Organic materials, such as banana peels, broke down completely, whereas synthetic polymers remained largely intact. Statistical analysis supported this pattern, indicating that substrate type is a strong predictor of degradation success. Waste pollution presents a significant environmental challenge worldwide, contributing to long-term ecological damage and public health concerns. But nature holds a solution: fungi. Oyster mushrooms are expert waste decomposers. This unique capability highlights the potential of fungi as an effective and sustainable tool for bioremediation, the process of using biological organisms to clean up contaminated environments. We asked the question, can oyster mushrooms biodegrade diverse substrates, and does substrate type dictate success? Our alternative hypothesis was that they can, and that substrate type does influence the outcome. In 2023, as a general biology lab student, I joined classmates in testing oyster mushrooms' bioremediation potential. Our team layered wetted cardboard strips serving as the growth medium into sterile jars. In my jar, I added dental floss. After inoculating all jars with oyster mushroom spawn, we sealed and incubated them at room temperature for 12 weeks. Through weekly observations, I documented my jar's progression. By week 3, pearly white mycelium engulfed the cardboard. By week 6, mycelium fully coated the jar's interior walls. By week 12, jars were opened and exposed to humidity and light to induce fruiting. By week 15, despite abundant mycelium and a fruiting body, the dental floss remained intact. In 2024, as a TA, I guided new students through the same protocol. This study analyzes a total of 112 trials across 2 academic years, including a diverse array of substrates such as natural fibers, synthetic polymers, and food waste. A chi-square test was used to quantify the substrate impact. Figure 1 on the left displays a student jar with full mycelial coverage on the interior walls, while the right shows the emergence of a pink fruiting body after week 12 exposure, characteristic of oyster mushroom. We found that fungi have food preferences. The table illustrates the effectiveness of substrate degradation, complementing the pattern shown in Figure 2. It includes specific examples for each category, such as cotton for natural fibers, which were moderately degraded, straws for synthetic polymers, which were minimally degraded, biodegradable plastics for composites and blends, which were minimally degraded, banana peels and food waste for organic materials, which were highly degraded, and marshmallows for the non-categorizable others, which were moderately degraded. This table demonstrates a wide range of outcomes across both organic and synthetic materials. The chi-squared test yielded a chi-squared value of 32.39 and a p-value less than 0.0001. Our results clearly show that substrate types significantly influence degradation outcomes, as confirmed by a p-value less than 0.0001. Organic waste materials were almost completely broken down, while plastics showed minimal degradation. This pattern reflects the enzyme specificity of oyster mushroom. An unexpected finding of this study was that oyster mushroom's ability to break down some plastics, a capability not commonly known. Some natural fibers and composite materials showed slight signs of decomposition, suggesting limited but possible potential for remediation. This lab successfully transformed student perspectives on fungi from overlooked organisms to powerful environmental tools, while directly exposing them to the complexity of waste breakdown and material persistence. Students experienced both firsthand the potential of biological solutions and the reality of global waste challenges, supporting UN Sustainable Development Goals 4 and 12, education and responsible consumption. Future educational iterations could include a wider variety of substrates and give students more control over the experimental design, deepening student understanding of biological diversity and environmental problem solving. In conclusion, this multi-year laboratory activity achieves its core educational objective, inspiring students to recognize fungi's environmental potential while building critical awareness of sustainable waste management. I'd like to thank you for your time, and a special thanks to Tatiana, Kuzmenko, Damien Willett for their guidance, and to the LMU General Biology Lab students for their contributions.